Hi, I'm Kate.
And I'm Augie.
Today we're going to be exploring different methods and ways that you can further explore what microbes are in fermented foods and drinks. Today we'll be exploring kombucha. Okay, so at this point you might have made a fermented food, seen some evidence of what's going on, but we still don't really actually have that much information about what microbes might be present in these samples. So today we're going to walk through a series of videos that help explain different methods and techniques that you can use to further understand what might be in here.
Yeast are found in kombucha samples, they're found all over our bodies, they're found in many fermented foods, and Saccharomyces is a budding yeast that basically ferments a lot of our favorite things. So a lot of people think about the gut microbiome and bacteria that colonize us, but there's also yeast in there, and they play a really important role in the way that our bodies function. I am a very big fan of yeast because I spent my time in graduate school basically studying how yeast that give us infections, Candida albicans, mutate to become resistant to the treatments that we have for them. I did my PhD at Emory University where I studied genetics and molecular biology, but more specifically now in my position, I develop labs for Harvard undergraduates to be able to explore biology in a different way.
Here we took samples from 3 different kombuchas.
Using our micropipette, we're going to take a small sample of the kombucha and add it to our tube. This is a micropipette, and they come in different sizes based on how much liquid you want to be transferring, but these are very, very common tools that are used in a scientific laboratory. They're very similar to the pipettes that you might be used to, except they're much more precise and a little bit pricier too. So I'm switching tips between each of these different kombucha because I don't want to cross-contaminate between them. So the next step is to take our sample to the centrifuge. This is a benchtop centrifuge, and it's used to spin tubes like this that have our mixture of kombucha really, really quickly. The purpose of centrifugation is to essentially separate a liquid into the solids, which will fall to the bottom, and then the liquids that will rise to the top. In our case, kombucha is made up of a bunch of different things, including microbes, tea, which is the liquid. So through the centrifugation, the microbes will form a pellet at the bottom, and then the rest will be the liquid layer on top. 2 really important things. The first is how much time, how long you're spinning something for, and the second is how fast, how quickly you're spinning it for. Sometimes when you spin it too fast, it can damage the cells, it can burst the cells, and we want as many cells alive as possible. 1 minute should do the trick, so we're gonna do that. Okay. Another important thing is whenever you're centrifuging, you have to make sure that the centrifuge is balanced. If it's not balanced, it can literally explode.
What do you mean by balanced?
So if you take a look in here, this one has 18 tubes, and what I mean by balanced is that there's equal distribution of weight across all of these axes, you can think of them. So that's why in this case, these 2 are across from each other, the 1 and the 10, and you can use these little lines here for guidance, right there. And then the 14 and the 6. So lock it in place. Alright, I'm gonna start it. So if it makes any loud noises, you know you need to stop it immediately. But this sounds really quiet, so we know we did a good job balancing. I'll show you. So this one was a pretty turbid kombucha, right? It was kind of dense.
Yeah.
And the store-bought wasn't. So that's why in this one, we see a pellet, like you can actually see a pellet. And in this one, you don't really see anything. The next step is to take out the excess liquid and leave the pellet, which forms during centrifugation. Then we add a little bit of saline water or salt water to help resuspend the pellet.